Thank you very much for your kind introduction. It's my pleasure to be here. People often complain about the weather in Bergen, but every time I come to Bergen and find it really beautiful weather, like today. So I apologize for the title of my talk that is full of acronyms, but I hope that at the end of my talk, you will be able to understand what these acronyms mean. Let me start from the geography. So I came from Vilnius, and Vilnius as a capital of Lithuania, and actually it's only 2 hours flight to Bergen. And I don't know how many of you have been in Vilnius before, but anyway, I should say that it's a nice city, and it's a UNESCO heritage site, and it's full of churches. And I find it that Vilnius probably has the largest number of churches per square meter in the world. But anyway, it doesn't mean that we go to pray to church every morning. Just across the river, there is a modern city, and in Vilnius history and modern times usually live together. And Vilnius University, and I belong to Vilnius University, is one of the oldest universities in the region, and very recently, actually, we moved into a brand new life science center. And my lab is located at the Life Science Center of Vilnius University. It's quite a big building that accommodates three different institutes. And in the vicinity of the Life Science Center, there is an incubator that hosts spin-off companies that originate from Vilnius University and also outside. And very recently, a Finnish company experimenting moved into university's incubator. And in 2021, we established a new research institute in Vilnius. It's Partnership Institute for Genome Editing Technologies, and this institute was established together with EMBL. And the goal of this institute is to discover, develop, and apply genome editing tools. And within this institute, we were able to hire 6 young principal investigators that came from different parts of the world. One of them came from University of Oslo. A couple of them came from US and Switzerland and other locations. So now I would like to switch to my scientific talk, and actually I would like to start you telling about immune system, but about immune system that is present in bacteria. So you know that in our body, immune system actually allows us to fight against invading viruses and microorganisms. So, and it has 2 different branches that are called innate immunity and adaptive immunity, and these 2 different branches by the cells and molecules that are involved in the manifestation of immunity. In the case of innate immunity, cells are able to fight against an invader immediately. However, in the case of adaptive immunity, the time is required to establish immunity. However, when immunity is established, so then during next round of infection, the response is quite quick. And actually, if our immune system fails, so then actually we lose our battle against parasites. But bacteria, including pathogenic ones, they also have their enemies. And phages or viruses of bacteria, they infect bacteria very often. And actually, phages infect bacteria in order to replicate. And the phage infection, in the case of lytic phage, is kind of question of life and death for bacteria, because phages represent a lethal threat for bacteria, but still bacteria somehow survive, and they survive because they were able to build different barriers that interfere with phage infection. And these defense barriers actually forces bacteria to build new, these phage infections actually forces bacteria to build new defense barriers, and then viruses are forced to evolve in order to break in through new defense barriers. And this is kind of arms race between viruses and bacteria that is going on and on. And there are multiple defense systems in bacteria, and in bacteria, genes that encode for different antiviral defense systems, they are clustered in a so-called defense islands in bacteria. And these defense genes are very important. For example, in bacteria, Microcystis aeruginosa, nearly 30% of the genome is actually taken by antiviral defense system. So this simply shows in what kind of hostile environment this bacteria lives, because it has to spend nearly 1/3 of its genome just to encode for different antiviral defense systems. So in my lab, we studied different antiviral systems in bacteria using a combination of biochemical, biophysical, and structural methods. So actually, why these antiviral systems are important? Because, of course, they are interesting, because actually they use very interesting biological mechanisms to fight against viruses. But there is also another thing, that actually these antiviral defense systems resulted in different technologies. For example, the antiviral defense system that is called restriction modification system resulted in recombinant DNA technology that enabled us to manipulate DNA. And in the same way, the CRISPR-Cas systems that I'll be talking later actually are behind the genome editing technology. So, and now there are hundreds of new systems identified. So the question is, what awaits us there? And the simple answer is, I don't know. Because actually, CRISPR-Cas system is a nice illustration. So we started our studies of CRISPR-Cas systems as antiviral and novel antiviral defense systems. And in order to understand how these CRISPR-Cas systems are able to provide interference against invading phages. So when phage infects bacteria, so most of the bacteria die. However, a small number of bacteria survive, and it turns out that the survivors were able to hijack small pieces of invading viral DNA and insert them as so-called spaces in a CRISPR region that is present in the bacterial genome. And then bacteria is able to use these pieces of viral DNA that are inserted in bacterial genome as templates to make small RNA molecules that are called CRISPR RNA. And these small RNA molecules, together with Cas proteins that are encoded by Cas genes that are located in the vicinity of CRISPR array, form a so-called effector complex. And this effector complex is able to destroy invading phage DNA during next round of infection. So in this respect, CRISPR-Cas system works as an adaptive immune system of bacteria, because it's able to memorize the invader by writing in invader's DNA into the genome, and then use this information in the subsequent round of infection to recognize the invader and to destroy the invader. So in 2012, actually, we have shown that one of these effector proteins that is called Cas9 and is present in a particular CRISPR-Cas system, in fact, functions as RNA programmable DNA endonuclease. So we have shown the Cas9 complex is assembled of two pieces. One of them is Cas9 protein that is bound to two two RNA molecules. And one of the RNA molecules that is present in a complex acts as a program that allows to actually deliver or address Cas9 to the target that is encoded by this CRISPR RNA. And then Cas9 protein cleaves both DNA strands at the target, introducing double-strand break. So that was a really fascinating system, because actually we showed that by changing this guide RNA, you can deliver Cas9 protein to any site in the genome, and then Cas9 protein will introduce double-strand break, and this double-strand break will trigger mechanisms of double-strand break repair that allows you to generate gene knock-ins and knockouts. Yes, so the specificity of the Cas9 protein is dictated by Watson-Crick interactions between the guide RNA and matching DNA strand. However, in order to initiate cleavage, the short nucleotide sequence that is called a protospace adjacent motif, or PAM, is required in the vicinity of the target site. And if there is no PAM, there is no DNA cleavage, even if the guide RNA perfectly matches the target sequence. And this PAM sequence is recognized by Cas9 protein, and different Cas9 proteins recognize different PAM sequences. So this is important thing, that in order to cleave DNA, Cas9 requires both matching DNA sequence and PAM sequence in the vicinity. So if you look at the and this discovery of Cas9 protein as a programmable nuclease was a turning point in a CRISPR history. So if you look at the CRISPR timeline, so BC, and BC stands here for before Cas9, actually, CRISPR-Cas system was just a peculiar antiviral defense system. However, after AC, it became a genome editing tool. And actually, it's just simply a nice illustration that there is a very thin line between basic research and applied research, because one day you are doing a basic research, next day you can be doing genome editing, and this is how things develop in science. Yes, so the Cas9 is a nice technology that is rapidly advancing into the clinics for treatment of different genetic diseases. However, it still has some limitations or challenges. I listed some of them in this picture, but today I would like to focus on one limitation. So it's called, oh, sorry, not this one. Yes, it's called delivery size limitation. So the Cas9 protein is a big protein, and the gene that encodes Cas9 is around 3,000 base pairs. So typically, adeno-associated viruses are used for delivery of Cas9 protein into the cells to do gene editing. However, the AAV has its own cargo packaging limit. So therefore, it's difficult to package large genes into AAV. So therefore, of course, you need to find, maybe, if you like to use AAV successfully, you need to find smaller genome editing tools. And of course, the question is where you can find them. And there are, of course, two approaches. You can try to engineer existing genome editing tools in order to make them smaller. But in my lab, we actually preferred a natural diversity approach that is probably nicely illustrated in this picture. So we decided to explore the natural diversity of CRISPR-Cas system that is really, really huge. And indeed, if you look at the existing CRISPR-Cas systems, they are very, very, very diverse. And they are subdivided into different classes. And in a class 2 CRISPR-Cas systems, the effector protein is a single protein that binds either two or single nucleic acid. And they are further subdivided into different types, like type 2 and type 5. And in type 2, the effector protein is, the signature protein is a Cas9 protein. As I said, it's a big protein from 900 to 1400 amino acid residues. And it has two different active sites, and each active site cuts a different DNA strand. In the type 5 family, and Cas12 proteins are actually effector proteins or signature protein in this case. So their size diversity is really big. So there are Cas12 proteins that are around 400 amino acid residues and Cas12 proteins that are close to 1,500. And differently from Cas9, Cas12 protein has a single active site, and this single active site has to cut both DNA strands. So we decided to look and focus on the diversity of the Cas12 family, and we decided to look at the Cas12 proteins that has a signed range within 450 550 amino acid residues. And a few years ago, Shmakov and Harrington identified a novel so-called CRISPR-5 type. And in this CRISPR-5 type, actually, the effector protein, at that time it was called Cas14a, but the nomenclature change is now called Cas12f. They identified effector proteins, and this was done bioinformatically, that were half size or even two sets smaller than existing Cas9 and Cas12 proteins. And they belong to the Cas12 nuclease family. They contained a single active site. And so they identified probably close to 25 different Cas12f variants. And we decided to look whether these Cas12f miniature nucleases are still able to cleave DNA and do genome editing. And we selected a bunch of Cas12 family nucleases in the phylogenetic tree. And we have a method that allows actually to characterize biochemical requirements for these miniature nucleases. And using this method, we were able to show that actually these miniature nucleases are able to introduce double-strand break, but they require different PAM sequences in the vicinity of the target site. So, of course, the next question was whether these miniature nucleases are able to indeed to cleave DNA in the test tube. We did a number of experiments that showed that indeed they are active in vitro. They cleave DNA outside the target sequence. But an interesting finding was that these miniature nucleases actually required increased temperatures for the cleavage activity. And if you look at this picture, so both of them actually required around 50 or 55 degrees Celsius for DNA cleavage. If you think about doing genome editing in human cells, it's probably not the best temperature to do that, because human cells doesn't like temperatures around 50 degrees Celsius. But nevertheless, we decided to look whether these miniature nucleases are active in eukaryotic cells, and we selected actually plant embryos, or more specifically, maize embryos. And one of the reasons is that maize embryos are able to tolerate short pulses of the heat shock. And actually, the nucleases were introduced into maize embryos using ballistic transformation. And then, actually, we tried to identify mutations that were introduced, hopefully, by Cas12f family nucleases in the maize cells using sequences. And what we found when the experiments were performed on two different target sites were performed at 37 degrees or 28 degrees, actually there were nearly no editing at these sites. However, when embryo cells were subjected for short pulses, temperature pulses, actually the editing efficiency increased significantly. And this shows that indeed the temperature is the major obstacle for genome editing in eukaryotic cells. So then the next question was, can we still do something in human cells? And we performed editing in human cells using Cas12, SP-Cas12F nuclease. And what we found, the experiments were performed in HEX cells. It's a model system in the lab. And what we found that indeed there were small edits at 2 different sites, but editing efficiency was really very low. And the reason probably is that actually the temperature was much lower than the optimal temperature of these miniature nucleases. But then, actually, our collaborators at Corteva company developed a method that allowed actually to improve cellular activity of miniature nucleases in a cell. And they engineered a protein library of miniature nucleases and used this protein library to target a specific gene in yeast cells. And in this case, if the miniature nuclease is able to cut this gene, so then the development of specific metabolites in yeast is disrupted and yeast cells turn red. So if you look at the wild-type protein, so actually there are not many red cells at 30 degrees, 37 degrees. There are few of them at 45. But then, actually, if you look at the protein library and if you look at the individual substitution in protein library, you see that in the library you can find yeast cells that are already, there are more red cells. And then if you combine all these individual substitutions, so in fact you can see that most of the yeast cells became red, indicating that probably the miniature nuclease is already active at 30 degrees Celsius. And of course the next step was just to take this miniature nuclease, engineered miniature nuclease, and try to do genome editing in human cells. And what we found that indeed this engineered miniature nuclease showed a really increased genome editing efficiency in human cells, indicating that actually probably the requirement for higher temperature was the major obstacle that resulted in low genome editing efficiency. So just to sum up this part of my talk, I showed you that these miniature Cas12F nucleases that are much smaller than Cas9 and Cas12 proteins can cut DNA in a time-dependent manner, and these miniature nucleases could be developed into genome editing tools that will be compatible and are compatible with AAV vectors. Because after our discovery, there were a couple of papers showing that these miniature nucleases could be transplanted into AAV vectors, and these AAV vectors could be used as delivery vehicles for miniature nucleases. So now I switch a bit gears, and I still would like to show you this phylogenetic tree of Cas12 family. And we just discussed these Cas12F miniature nucleases. But if you look at the top of this phylogenetic tree, so there is kind of one branch that stands for TnpB. And TnpB has nothing, this is another acronym, but it has nothing with Cas proteins or CRISPR-Cas systems. But it's still on the branch of the Cas12 family nucleases. And these TnpB proteins are really, really small ones. It's about 400 amino acid residues. So where this TnpB comes from, and TnpB is actually a protein that belongs to the transposon, to a specific transposon family. And transposons are mobile genetic elements that jump between different locations in the DNA. And this specific transposon that belongs to this family, and we focused on the transposon that belongs to the Deinococcus radiodurans bacteria. It's a very simple transposon. It's comprised often from single or two genes that are flanked by left and right end sequences. And then, actually, in the vicinity of the transposon, there is a specific TTGAT sequence in this case. That is the site where transposon is excised or inserted. When transposon actually travels, it looks for this TTGAT sequence and then inserts at this specific sequence. And it was well established that TnpA is a transposase that enables mobility. So transposase excise transposon, and then transposon travels to different location. But it was not clear what TnpB is doing, that TnpB protein is doing in this transposon. And the transposition of this mobile genetic element actually happens during DNA replication. And transposon is excised from the lagging DNA strand. And then, actually, when transposon is excised, so then it tries to find another location at TTGAT site and insert into the genome. And the transposon is excised as single-stranded secular DNA intermediate. And this is actually how this transposon travels. So we aim to establish what is the role of TnpB in transposition. And we used the similar approach that we often use in my lab. So we decided, let's try to isolate TnpB protein and characterize it biochemically. And what we did, that actually we were able to isolate TnpB protein in a heterologous E. coli host, and it turned out that TnpB co-purified together with RNA molecule. And because we isolated this nucleic acid from TnpB complex and then subjected it to DNase or RNase cleavage, so DNase had no effect on the cleavage of this nucleic acid, but RNase, it was sensitive to RNase cleavage. So the next obvious question was actually, where this RNA comes from? And we isolated this RNA and subjected it to RNA-seq, and what we found that RNA that is present in TnpB complex, oh, sorry about that. The RNA that is present in TnpB complex actually originates from mRNA that encodes the same TnpB molecule. So the RNA matched 3' end of mRNA encoding TnpB protein and also included right transposon element and 16 nucleotide variable sequence that belong to the plasmid vector. Since it included the right transposon element, we call this RNA like RNA molecule. So then the next obvious question was whether this TnpB protein that carries RNA is in fact able to cleave DNA similar to CRISPR-Cas nucleases. And we again had a simple biochemical screen that reported that indeed TnpB is able to cleave DNA introducing double-strand break, but only when a TTGAT site is present in the vicinity of the matching target sequence. But if you look at the sequence of the site that licenses TnpB cleavage, it exactly coincides with the TTGAT sequence that is required for transposon mobilization. So therefore, we call this sequence a transposon-associated motif or TAM. So next, taken together, all our data shows that TnpB protein, in fact, is RNA-guided DNA endonuclease. And yeah, probably due to the lack of time, I will not go into the details how it works. But anyway, we proposed that, in fact, the TnpB recognizes the target site that is a composite. One part of this target site is recognized by the RNA that is present in TnpB, and another part of recognition site is recognized by TnpB protein. So when transposon is present, the target site for TnpB protein is interspaced by the transposon, and TnpB is unable to cleave DNA. However, when transposon is excised, so then the TnpB target site is reconstituted, and TnpB is able to introduce double-strand break into the DNA. And then what happened next? So then in the cell, mechanisms of the double-strand break repair or homology recombination results in the recombination on the TnpB, recombination of DNA molecule that lacks transposon that was excised. And then actually this another transposon copy is reinstated after transposon excision. So this mechanism actually helps to secure that transposon is never lost in the cell. So of course, next we wanted to look whether this TnpB protein could be harnessed as DNA editor in human cells, and indeed we found it is the case that TnpB is able to cut DNA introducing indels at 3 or 4 different target sites with different efficiency, but this 20% efficiency at one of the target sites is very promising because it was done without any modifications. So next what we did, we decided to look how this TnpB complex looks like, and we were able to solve the binary and ternary structure of TnpB complex using cryo-EM facilities that are present at the Life Science Center. And indeed what we found that the RNA structure in this complex is very complex, and it has really complicated secondary structure, but nevertheless, the cryo-EM structure of TnpB allows us to think about rational engineering of guide RNA or TnpB molecule just to improve the genome editing efficiency. And next what we did, we decided to compare the TnpB structure with structures of different proteins that belong to Cas12 family. And this structural comparison actually revealed that TnpB is the smallest or minimal core of the Cas12 nuclease family. So the question is that, is it true that then TnpB was a progenitor of the Cas12 family and CRISPR-Cas systems? And indeed, it looks that it's true because we also managed to compare TnpB sequences, and TnpB sequence is really widespread. There are millions of different TnpB sequences in the databases. And what we found that indeed the ISDRA2 TnpB that we characterized sits in the center of TnpB universe. And then actually many Cas12 family members form sub-universes, but they are much less populated. And this suggests that TnpB probably was the progenitor of the Cas12 nuclease family. And this is kind of a bit a paradox because TnpB is a mobile genetic element. But then actually CRISPR-Cas systems was able to tame the mobile genetic element and use it to fight against other mobile genetic elements like phages. So this shows how actually evolution is happening, and that some functional units are just used like guns for hire, so everyone can take if they are able to tame it. So this brings me to my summary slide. So I showed you today that actually we were able by looking at the natural diversity of the CRISPR-Cas system and TnpBs, we were able to expand the CRISPR-Cas toolbox, and now we have CRISPR-Cas tools or TnpB tools that are much smaller, and we think that all these tools could be harnessed for very different genome modifications. And finally, I would like to thank the people who contributed to this work, people in my lab at Vilnius University. We have established a company called Caszyme at Vilnius. It's located in this incubator, and they actually making all these gene editing tools that we discovered available for research community. And I would like to thank our collaborators at Corteva, New England Biolabs, and also Novo Nordisk Foundation Center for Protein Research. And thank you very much for your attention.